Good evening students. Today we will see the physiology of muscle contraction. So the skeletal muscle consists of two filaments, thin and thick filaments. The thin filament is made up of actin, troponin and tropomyosin, and the thick filament is made up of myosin. Here the actin and myosin are called contractile proteins, and troponin and tropomyosin are called regulatory proteins. And arrangement of these filaments gives a striated appearance, and under electron microscope these filaments forms bands. There is a darker A band formed by thick and thin filament. In the central portion there is no thin filament, that is a lighter H zone. On either side of the A band we have an isotropic band, that is I band. The I band is made up of only thin filaments, and these thin filaments attach to the Z line, and the distance between the two Z line is called sarcomere. It is a structural and functional unit of the skeletal muscle. The resting sarcomere length is 2.5 microns. With this introduction, we go for the second point. What is excitation? Excitation of the skeletal muscle is through the nerve. So when the action potential travels through the nerve, it reaches a neuromuscular junction where it causes the influx of calcium, and that causes the release of acetylcholine from the vesicles, and this neurotransmitter attaches with the acetylcholine receptor in the muscle membrane, and this receptors are ligand gated, and they contain the sodium channels, and this sodium channel gets opened when the acetylcholine combines with the receptor, and this causes the motor end plate potential, and these motor end plate potentials are converted into action potential, and the action potential travels through the sarcolemma. This is the process of excitation. Now the third point is how the excitation contraction coupling takes place. Now this action potential which is traveling in the sarcolemma enters the T tubule, and it reaches the receptor in the T tubule, which is called dihydropyridine receptor, and activation of this causes the activation of another receptor in the L tubule, systole of the L tubule, which is called the ryanodine receptor. The ryanodine receptor are nothing but calcium release channels, and calcium is released from the systole into the sarcoplasm. This forms a very important point of excitation contraction coupling. Now the third point is how the contraction takes place. This was explained by the sliding filament theory, or Huxley theory, or ratchet theory, or cross bridge cycling theory. What the theory says is, so the calcium which is released from the systole now combines with the troponin in the thin filament, and this moves the tropomyosin away from the myosin binding sites. So this tropomyosin will be covering the myosin binding sites in the actin filament. So once they are uncovered, the myosin binding sites are exposed in the actin filament. Now the myosin head attaches with the actin filament, and it will pull the actin filament. So what is taking place in the myosin part is the ATP, which is attached with the myosin head, it is cleaved into ADP and phosphate, and after this cleavage the head gets attached to the actin filament, and there is a release of phosphate, the next step, and this causes the bending of the neck of the myosin, and this is called the power stroke. When the neck bends, the actin filaments are pulled, and this forms a very important step in the muscle contraction. Now how the relaxation takes place, that is the fourth point. Now after the pulling of the actin filament, new ATP will attach with the myosin head. When a new ATP attaches, the myosin head get detached from the actin, and that causes the relaxation, and the calcium from the troponin will be taken back by the systole. So these are the two steps that forms the relaxation. The fifth point is applied aspects. There are three applied aspects in muscle. Number one is rigor mortis, number two is myasthenia gravis, number three is Duchenne muscular dystrophy. So rigor mortis is stiffening of the muscle due to the lack of ATP, and this causes the muscle is unable to relax because of the lack of ATP. The myosin head is not able to detach from the actin filament. Second is myasthenia gravis. It is an autoimmune disease where antibodies are produced against the acetylcholine receptor in the neuromuscular junction, and that causes the block in the neuromuscular transmission. And third is Duchenne muscular dystrophy, that is mutation in the dystrophin protein that causes muscle weakness. So these are the three applied aspects. Thank you.